recording. So yes, May the 4th be with everybody. And we are going to be going over the review page here for exam 2. Number 1 tells you to have a general understanding of Mendel's laws of segregation, independent assortment, and how it relates to the biology of cell division, specifically for chromosome division. So, first things we're going to talk about is that we need to know the difference in mitosis and meiosis. And let's see, get a whiteboard. All right. So, mitosis. I don't know why we're in purple, but whatever. All right. Mitosis is regular cell division. And this is giving exact copies of the cells. Two daughter cells. All right. So when we have mitosis occurring, because you're getting exact copies of DNA going to the daughter cells, there's no crossing over. There's nothing to worry about with it. By comparison, we have meiosis. And this is where we make sex gametes. This is your eggs, your sperm, and we get unique cells. Unique haploid cells at that. They're going to remember haploid is having half of the number of chromosomes, and then when you meet up with the other egg or sperm, you end up with the diploid cell. So these are unique haploid cells, and we have crossing over happening. Now, in regards to Mendel's rules, we don't need to worry too much about them, except to remember when the cells divide, this is going to be our cell here, and you have metaphase 1 and 2, but you don't necessarily need to know all the phases. I'm just reminding you that where they line up along the metaphase plate here, ooh, that's a wonky one, is completely random. You could have this, or you could have any of these pairs flip-flopped in the middle to have the other one on the left and the blue on the left and the pink now put to the right. So you get mixing of genetic information just from how they line up on the metaphase plate. And this is where if your genes are on completely separate chromosomes or are very far apart, you're getting stuff segregating independently. Now, because Mendel did not discover crossing over, because all of the chromosomes or all of the genes he was looking at were on chromosomes that were either separate or on separate chromosomes, or they were very far apart. On human chromosomes, we have several crossovers that occur on each chromosome. And so if you had them at two opposite ends of one chromosome, you're going to pretty much guarantee that there's going to be some crossing over action happening between them. So they're going to be assorting independently. Then we end up with mutations that happen to our chromosome numbers by, let's say these go to divide during chromosome 1 and now, or during meiosis 1, and then we go on to meiosis 2, actually, let's see. And this is just following one set of the homologous pair. And because we lined up on this side, all of the blues went over here. Now remember that these get attached to the meiotic spindle. They attach at their centromeres, their spindle. and they're going to get pulled apart during anaphase. Now, if one of these doesn't get pulled properly, what we normally we would end up with one of the daughter cells being, can't go down, so I'm gonna have to go over here. These are our 2 daughter cells. And we should have half of it. But on this one, we got an uneven pull. And both of the attached sister chromatids came to one of the daughter cells, and the other one got shortchanged. So now if this one goes and meets up with another egg or sperm, it's going to end up with 3 copies instead. So this is where you get things like Down syndrome, where you have 3 copies of chromosome 21. And there's things like Turner syndrome that was only 1 copy of the X chromosome instead of the normal 2. Klinefelter syndrome was 2 copies of the X and a Y. There's lots of different sex chromosome mutations you can have. And pretty much any of the other autosomal chromosomes, if you have 3 copies, you don't end up with a viable offspring. They die in utero. So sex chromosomes and Down syndrome are pretty much the only ones. I won't ask you to memorize the names of specific disorders. If there's something about this, I would say like in parentheses and give you information about the disorder that I'm asking about. So if I say Klinefelter syndrome, and then I'll put in parentheses XXY so that you know what it is that I'm talking about. Because it's not a medical class. I don't want you getting too hung up trying to memorize a bunch of disorders. They're just in there to give you an example that you might remember from your normal life. Is my fish tank too loud for anyone? Is that getting any problems, or can everybody still hear me? Nice.
I can hear you. I don't hear a fish tank.
Okay.
Yeah, it's fine.
All right, cool. I tried to move away from it, but it's bubbling. So, okay, so this is pretty much everything you needed to know for number 1. Let's close that out.
Sure, blame it on the fish tank.
Oh, the fish tank bubbling in the background is, fish tank, but it's the filter that runs on top of it. And I realized earlier the mute button being on was my son was using my computer for Zoom meetings earlier with his class and apparently got muted. And I never pressed that button, so I didn't realize it was on. All right. Okay. I do want to remind you now, if we have a mutation that occurs, like the one I showed in the sex gametes there during meiosis, and it goes on, it's going to then go into the zygote and through every single cell of the individual as it grows. If you have a mutation that occurs later on from mitosis, it's only going to go to the daughter cells that originated in that one. So, like, for example, growing up and you get a bunch of sunburns and now the mole on your arm starts changing and becomes cancerous. That cancer doesn't go immediately into every cell in your body. It's only going to the ones that are formed from the cell divisions from that one that got mutated. So if it occurs during mitosis, it's only going to affect some cells in the body. If it occurred during meiosis, it's going to affect the cells, every cell in an individual. But that also means if you want to do some kind of gene therapy, if you need to get it into every cell of the body, you have to have it very, very early on in embryo formation, or it has to be something that doesn't need to affect every cell. So I believe the book talked about cystic fibrosis that affects the lungs. And in that case, they can do, and they've been trying some gene therapies that they can just inhale the vector. So by inhaling it through an inhaler, it's going into the lungs, but it doesn't matter if your toes aren't cured because your toes aren't causing any of the symptoms that you're seeing. So just think about that in terms of mutations and what parts of the body are going to be affected then. Number 2 is Punnett squares and probabilities for offspring. You should be fairly familiar with this by now. It's just going to be basic Punnett squares. It won't be any more complicated. This is just kind of a callback to the first test. I will show you one that might be a little more complicated. Okay, so let's clear.
For the exam, are we gonna be making a Punnett square?
If you might need to for some questions. That's why I'm going to give y'all kind of an example one here. Just for a more complicated one, I'm going to show you a sex-linked one, since that's something that you should be pretty familiar with, the basic dominant and recessive. It just might be that I ask like a question that says, what is the probability of the offspring being a certain type? And you might need to doodle your own Punnett square to figure out the ratios. Okay, so because the test is going to be online, I'm limited by what I can ask. So it has to be something that you wouldn't be expected to be able to draw on there, kind of nature of the beast. But all right, so we have a Punnett square. So let's say we want to do something like colorblindness. So colorblindness, if you don't remember, is X-linked recessive. Mom has 2 X chromosomes. Dad's got the X and the Y. So let's say the information given in the question is that you have a colorblind dad, and let's see, we'll go with this one is colorblind. B. And mom has normal vision. And we ask, what are the odds that a son born from these parents is going to be colorblind? So you have the option then that you can draw out everything. X, Y. That's little b.
Wait, I have a question.
Uh-huh.
Wouldn't that mean that there's no possible way for the son to get colorblind because he would have to get the recessive gene from his mom, and he can't get his X chromosome from his dad?
Then you would say it's 0%.
Oh, okay. So you don't even have to necessarily do that score for something like this specific.
No, you don't need to if you don't need to, basically. Yeah, I was just going to draw it out there just for a reminder on how to do Punnett squares. And then I'm going to also talk about what are the odds if it's a daughter then. So, so if it's a daughter, we know, we can see, sorry, I have to finish all of these. Dad gave his here and here. And mom gave hers here. Here. Sorry, I'm having to use like the little touchpad mouse. All right, so as she pointed out here, so for a son, he can't get the recessive version from dad because all of his X chromosomes are going to come from mom. So 0% chance of a son having any color deficiency. Now for the daughters, they are going to have normal color vision, but they are carriers now. They could potentially have colorblind offspring. As we said, in this case, colorblindness is recessive. So I could give you any kind of variation on this type of question on a test that would say, you know, if the mom is a carrier, or we don't know if mom is a carrier. We know she can see color vision, but just because if you're a carrier, you can see color, but you could still be carrying this hidden recessive trait.
So if you were to ask a question on the test like, we don't know what the parent, like the mom specifically has, but the son is colorblind. We can assume that the mom has a trait for the recessive gene.
Yes,
right? Okay.
exactly. Yeah, it would be given that you have something just like that, where dad has normal color vision, mom has normal color vision, son is born with color blindness, and what is the genotype of mom?
Oh,
And
okay.
so you could, or if his sister is born, what are the odds that she'll be colorblind? You know, something like that.
Okay.
So,
Cool.
yeah, so you might need to draw out a Punnett square just to make sure you're understanding the question, but there won't be any, I don't think there's going to be anything expecting you to draw it out, because I won't be able to see your drawings the way if you were taking the test on paper. The only other thing I could think of would be maybe if there was like a blank here and said, what would go in this blank? And then you have like a multiple choice or something like that you would be able to complete. But it's going to be questions done online. So. And just like the first test, there's going to be some multiple choice, some short answer, some like true, false, and some of the questions from the first test are versions of them will make a comeback. Now, I'm not going, while you should still remember how transcription and translation work, it's going to, any questions about that will be more basic knowledge of, you know, things you should still feel comfortable with. So if it pops up again on the study guides, that's why. It's the kind of like, hey, I've seen this from before. Yes, because we just built off the ideas. Dominant and recessive don't just go away because we start talking about incomplete dominance. So, all right, so that was number 2. But that does bring up now modes of inheritance for number 3 here. This includes extensions of Mendelian genetics like incomplete dominance, sex-linked, lethal, etc. And tells you to practice examples like blood typing and hemophilia. We like blood typing as an example because it's a codominant that's easy to see. Let's see. Going back here to my whiteboard. Here the drawings. So remember, for blood typing, we use capital I, and big A is dominant, big B is dominant, and then we have lowercase I for type O. So if somebody is big A, we'd say they have type A blood. If they are big B, they're type B blood. If they are AB, they are type AB blood. It's codominance. These are like the enemy flags. So you can have both of them present. The recessive one is no flags. There's nothing to mark for O. So if somebody is type A and has that recessive I, their blood still looks like type A because there's A flags hanging on it. There's not a type O flag. That's just saying nothing. So you have to have the double type O, 2 recessives, to be able to show a type O blood. If A or B is present, they are both, they are going to overpower the recessiveness. Now, I do kind of want to talk about dominant recessive from a molecular standpoint, because in this case, a blood cell having the antigens on the outside of it, switch colors here. Let's say that the A's have little points on it. It doesn't matter how many little points are on the cell. As long as you have one copy of the A gene, you're going to have the points on your cell, these antigens. So being big A doesn't matter compared to being big A, little I. All right. So when that happens, when you have complete dominance, it means that having just one copy of the gene is enough to make it appear 100%. that makes sense. Or for it to be a fully functional protein. One copy makes 100% of what you need. So if we think about a flower here, switch over and say, on the flower, you can have red, and then, or the flower can be white. So when we show red is dominant to the white here. If this somebody is RR, you end up with the white flower. If something is completely dominant, like the way Mendel saw it, when you have that one R, that is going to be enough to get a red flower. One copy of this gene is enough to get 100% pigment. Having two copies doesn't make it get any more red. And this is the type of thing that Mendel thought he saw, which actually they go into more detail, and they found out if you get down and like look at this like wrinkled versus smooth at a microscopic level, it turns out there was some incomplete dominance going on, but not enough that you could see with your naked eye. So you don't have to worry about it. But if the homozygous dominant and a heterozygous look exactly the same, that's complete dominance. Incomplete dominance would say that this heterozygote in the middle here is now pink. That means that each of the big R genes is only giving 50% of the color. So you had to have two copies to get to 100%, versus on the heterozygote, having one copy just gave you 50% color. 50%. Where up here you had 50% plus 50%. Right? So this is incomplete dominance. Incomplete. I probably could have typed this better. Incomplete dominance. This one over here, if you end up with both of them showing on the outside, like this is co-dominance. They are co-captains. Now, if your flower, instead of coming out pink, was like splotches of white and splotches of red, then you could say that they were co-dominants. It just depends on what you're looking at. White is usually a weird one to see that with. You might have, like, you could think about like dogs that have multiple hair colors, and those can be co-dominant to each other. So you have the two genes, and did you end up with a gray puppy, a black and white dog, and then you ended up with a gray puppy, or did you end up with a spotted puppy? So, all right. The other thing in terms of Punnett squares that could come up is lethal. Oh, actually, back. And the lethal mutations are, yeah, you could have lethal dominant and lethal recessive. The one that talked about in the book was the Manx cat. And I'm actually this way. And here, if you had a normal cat and a carrier, or sorry, two carriers, big M and little m. And the mutation to get the tailless is dominant, the capital M. So actually, since these are dominant mutation, both of these are two tailless cats then. That looks weird. And middle is normal. So then what they see is we do the normal Punnett square. We get our big M, little m over here. Big M, little m, little m. And they go, all right, cool. But what happens is the babies are born and you end up with two tailless and one normal. So what happened to get a ratio of 3? Look up here. This is the lethal mutation. Let me swap colors. If you had the double dominant, it died in utero. So you have to be a heterozygote to be able to show, because otherwise you were dead. And then this kitty is normal. So anytime you see a ratio that it's not out of 4, like, you know, 1/2 or 1/4 or something with an even number, if you end up with 2/3, that tells you something's funky. And you probably want to look at something going, where did my other square go if I only have 3 showing? So chances are that there was a lethal mutation involved if you end up with thirds of anything. There is the exception where you have like penetrance, but we didn't go into that very much for penetrance and expressivity. Incomplete penetrance does happen where sometimes you end up, like, let's say you have a heterozygote kitty here where one of them has part of a tail, or some of the, not all of the cats that are big M, little m are tailless. You could have some that if only 70% of it, then you would say that the gene had 70% penetrance. We don't talk about that very much in this class. So, I mean, it can happen, but if you're getting nice, clean 1/3 or 2/3, then that's probably not a penetrance question. That's probably something like this with the lethal mutation. So, lethal. Okay. Does anybody have any questions over these before I go on? We good? Chat's still good. Okay. Can y'all still hear me?
Yes.
Okay, like in silence. All right. Let's see. Clear drawings. Next one. All right. And hemophilia is just a sex-linked one. Since I already talked about sex-linked with colorblindness, it's the same idea. Just got to remember, is dad's only giving X chromosomes and all the Y's have to come through dad. Dad is only giving 1 X, mom gives 2 X. And so if it's X-linked dominant or if it's X-linked recessive.
I have a question. Is there going to be anything over the heterozygous advantage, like with sickle cell, or is that just going to? We didn't really go over that too much.
I mean, that's kind of the only example that we ever really talk about with it. I mean, you sometimes, the only thing other than that I could think about is when you end up with dogs that are mutts, that the more options you have, generally the better, the more physically fit the offspring is going to be. But no, we don't. I'd have to go back and check. If there is something, it might just be a question that's literally just saying, what is it called when heterozygote is, better off than the
I get recessive or dominant.
than either of the homozygous versions. Yeah. And it was literally just a vocabulary word. But yeah, we didn't go into any real detail on that. So,
Okie
all right. Pedigrees. This is, I say, includes extensions again. I mean that now we can have some sex-linked ones in there. When we did exam 1, we only had regular dominants and recessives. There weren't any sex-linked genes on there. We talked about this with the wolf hair one, which also pulled up hemophilia. If you just Google pedigree practice problems, there's lots of websites out there that have little online ones if you want to look at it. So you don't have to worry too much, just basic pedigrees. If you had trouble on it before, just practice on them and get some review in there if it's been a while. But it's going to be the same idea as that I show you a pedigree and ask you, all right, figure out the mode of inheritance. And you have to look at all the offspring and be like, okay, that's a dad passing it to all the daughters and none of the sons. Then we go, okay, then it's probably sex-linked, something like that. And then on sex-linked inheritance, we didn't talk too much about maternal effects and imprinting. Just know the examples the book gives is for methylation and how sometimes during embryonic development, all of the methyl groups will get wiped off of some of the genes, and then they'll get put down to match whatever parent version, like it'll get switched over to only whatever dad gave or only whatever mom gave. That is something that occurs. There's some growth factors that have that. Maternal effect, I don't even know if it's in this edition of the book, is just saying that the proteins that come from the mom as the baby's developing is going to influence what the baby's phenotype is. So they have these cone snails that can swirl to the left or to the right. It doesn't matter what the genetics of the baby are. It mattered what the mom was. And because her nurse cells were giving the developmental hormones, if she is handing out hormones that say turn right, the shell is going to turn right, even if the baby's DNA says turn left. Then when that baby has offspring, it's going to be pumping out turn left proteins. And now its offspring are going to turn left, regardless of what their DNA says. That's all maternal effect is. Technically, you can also kind of throw in mitochondrial diseases. We didn't really talk about those very much, but there are some diseases in the world that it's not the genomic DNA that's broken, it's the or it's not the nuclear DNA that's broken, it's the mitochondrial DNA, and you end up with disorders in the offspring that can be pretty bad, because if the cells don't have enough energy, they usually don't survive. So gene organization, so this was just what was on there from last time. Promoter, operator, coding regions, etc. Don't memorize the specific sequences, like don't go TA, TA, TTC, that kind of thing. But you should know the general idea of how a gene is set up. So let's see. For the reminder, there we go. Gonna have our DNA here, which I can talk about the Lac Operon as our example here. Our promoter, I'll do it this way and make promoter green, because green means go. Okay. Promoter, P-R-O. And then the rest of the gene, like this, we'll just say gene. All right, so promoter is always upstream of the gene. The promoter is where all of the machinery assembles to be able to go forward. Now, part of the promoter here is a region called the operator site. And the operator site is where the repressor can bind. All right, well, let me add the S. Okay. So if your repressor was going to come in and block things off, this is where he's going to do it. Now, even if you assemble all of the machinery here at the promoter, that repressor is in the way. That repressor. All right. Now, in the case of, there's a story that I usually have my class act out in the classroom, and everybody wears their name tags and works through the Lac Operon problems. We didn't have that option this time. So you can, the whole story is that glucose is basically a kingpin mafia boss that runs the city, and it keeps everything shut down as long as glucose is in control. There is a gene called beta-galactosidase that's what helps break down lactose into its subparts, allolactose, that can bind to the repressor. Now, beta-galactosidase is a shut-in because she doesn't get any visitors. The repressor is like the muscle for the mafia. He's stands there at the door as security, and this is beta-galactosidase's apartment complex. So the promoter for the apartment complex can stand out front yelling, "Come on in," all they want, but if you can't get past security, you're stuck there. The operator is our door person, who's like, well, do I let people in? And the repressor is keeping the operator silenced. She can't do her job because they can't get past the doorman. Now, whenever then Lactose's freedom fighters come in and they bind allolactose here, lactose in the environment, binding here to the repressor and making it so it can no longer stick, then you end up with the repressor no longer stopping it. Now ribosome can show up and gene transcription can occur. And in our story, sigma factor, who was the little subunit that helps the ribosome find the promoter to know where to start, is the Uber driver. So sigma factor drops ribosome off at the door to the apartment complex and says, go on in, everything's fine. So doesn't matter if your sigma factor's there if you got a door person standing in the way. So as long as the allolactose is there to bind to the repressor, repressor can be shut down. The freedom fighters have overcome. In the meantime, you have another place further upstream that, in our story, we have it as a tree house. This is the cap binding site. So the cap binding site is upstream of the promoter, and adenylate cyclase creates cyclic AMP, and cyclic AMP and cap all get together and they bind, and these are activators. So add it in. And you can think about this as if you were debating to move into an apartment complex and they had a cool tree house next door with kids playing, you're going to go, oh, this is a nice place to live. I'm going to go ahead and come on into this apartment complex. So it doesn't matter. You can have all the kids playing happily in the yard as you want. If there's still a security guard here to block it, you can't get into the apartment complex. So the level of glucose in the environment controls the amount of kids that are playing outside, if that makes sense. Let me type that out. Glucose present equals low cyclic AMP. Glucose absent equals high cyclic AMP. And remember, cyclic AMP is an activator. So whenever the mafia lord's gone, all the kids can run out and play. In our story, a Denolite cyclist was a merchant and cyclic AMP was her offspring. And they could play with Cap and Camp. Cap and Camp could get together and play at the treehouse as long as glucose wasn't around. Now, for the repressor, so glucose is being there is going to keep cyclic AMP down. So that means that if lactose is present, here's another option here. So lactose present equals no repressor. The allolactose binds to the repressor. Lactose absent is the repressor is bound. So this is the way that the cell controls. As long as glucose is present, it doesn't matter if lactose is there, because you don't get very many people showing up at an apartment complex that looks really inhospitable compared to when it's glucose is absent. Now it's like putting giant flashing neon lights outside that say, yay, come on in, everything's good. So if glucose is present, it doesn't matter whether or not the repressor's in place. You're not going to get transcription turned on very well unless you have that activator there. So when glucose is gone, we have an activator. It's going to say, hey, I'm starving here. I don't have any glucose. I need you to find another food source. And that's when it goes, okay, let's start breaking down lactose. Lactose is harder to break down and less energetically favorable than glucose. Glucose can go straight into the metabolism cycles and be able to be broken down for the maximum amount of ATP. So the cell is going to use the easiest form of energy currency to get what it wants. If that easy form of glucose isn't available, then it will use lactose. But you should know general ideas of enhancer regions, silencer regions, all of that's usually upstream of the gene, because it's going to be things that will either physically block the ribosome from being able to, or not the ribosome, physically block the machinery for transcription from binding there, or they are something that's going to bend the DNA around so the DNA is blocking. It was the mediator that acts as a go-between, but you don't have to worry about any of the specifics on things like that. Okay. Any questions? Are we good on Lac Operon and general gene?
I have a question. So when there's gonna be questions over this on the exam, are you gonna ask us to write out this whole like DNA strand where it shows where I call them camp activators because that's what I thought it was pronounced. Where you're gonna have to like write where the activators are and the operators and the promoter and the gene. Or are you gonna ask more like, here's a worded question, there's glucose present, what does that mean for the activity of, the CM?
Yeah, it's going to be more like that it's going to be questions about it, or saying, The promoter is upstream or downstream of the gene.
Oh, okay, cool.
Because you're taking the test online, I can't really have you draw things out very well.
Oh, yeah, that makes sense. Okay, yeah.
So most of the stuff is going to be either drag and drop
Mhm.
into place, or like I might give a true false question. But it's just kind of remember what a promoter is, what it does. Promoter is going to be where your machinery is dropping, but then you get your mRNA, and the mRNA goes off to the ribosome. So polymerase, there's the word, I could not think of that, sorry. Polymerase will start at the promoter region, and then your mRNA that comes out our mRNA here. And remember, the mRNA has the 5' cap and the 3' poly A tail. Well, actually, no way. If it's coming out this way, whatever. I kind of drew it backwards. Here's his cap. Here's his tail.
So would it be the other way around?
Yeah, I was gonna say it depends. I drew it kind of like it depends. If our polymerase were here, the part that came off first, this one's totally done. He's free floating, so his direction doesn't matter. Just remember, it's usually a 5' cap, 3' tail. Poly A tail. I was going to show that it was going to leave, and then it would have to move out of the nucleus completely before the ribosome could attach, but I kind of ran out of room. So then ribosome attaches here and then moves along. Ribosome. Terrible mouse writing. Ribosome. All right. So just always remember that there's some in between. You have a promoter, then you have some in between space before the gene starts. The gene does not start right here with ATG, because then there wouldn't be room for the ribosome to attach. ATG is going to kick in, I don't have room, further like down here. I guess over here it'll be AUG. Instead you give it like down here. Why can't erase that? Apparently. Erasing all my other stuff. Remember, it does not start immediately ATG after the promoter. It has to have some untranslated regions in between. We called it UTR. And they will be transcribed, but they won't be translated. They're going to be what the ribosome's going to go, don't care, don't care, don't care. Oh, there's my start site. Where's my methionine to initiate? And technically the ATG is written on the other strand, so it's actually backwards. I'm not worried about it. I just want you to understand the locations of these. So yeah, you won't be expected to draw it out, but you would be expected to understand what it is. Operator site is usually part of the promoter region. I know in the book when they talked about the Lac repressor, they showed that there are actually like 3 different operator sites. It's just what is the sequence that the repressor could bind to.
I'm sorry, can you ask that again?
The operator sites are usually part of the promoter region. That's just the name for any sequences that the repressor can bind to. There's actually like 3 different operator sites that they talk about in the book. I don't expect you to know. It's because the Lac repressor has like this U kind of shape. And so it has to physically grip the DNA, and it'll like twist the DNA around to make it to where the polymerase can't attach to it anywhere. If it's all knotted up, it's like, I don't have anything to start reading here. And that's how the repressor is keeping it silenced.
So the repressors are what's basically restricting a bunch of enzymes, right?
Mm-hmm. Yeah, the repressors are physically sticking to the DNA to keep the polymerase from being able to assemble all of its machinery and do its job for transcription.
Okay. And then, so the Lac Operon, that's a repressor, or that's an operator, because I was a little confused on that.
Okay, Lac Operon. Operon means that there is one promoter and then several genes that will get transcribed. So let's say they have like gene Z, gene Y, like that kind of idea. That is an operon. So
Oh, okay.
promoter turning on a bunch of genes. There's actually several genes here in a row. It keeps going.
And so in between these genes, are there operators between them? Are there promoters between them? Or they're all only connected to one promoter? It's
By definition, an operon only has one promoter.
Okay.
That is just literally what it means, is one promoter turning on several genes. This would be transcribed as one big mRNA that could then be cut apart later. So there might be some junk DNA in here in between, but the polymerase isn't going to stop and then reattach. It goes all the way through to turn them all on or none of them on. I mean, occasionally you might get something that makes it fall off, but it's not like you're going to get the first gene and then another repressor is going to bind down here. No, it doesn't work like that. Operators you have to be up at the beginning to stop things before they begin. Usually, I mean, I say that and there's going to be something weird, but the way that they talk about it in the book and what you see is that the repressor usually is binding to something that's going to physically block the promoter region from being read. So if you see, like with mediators and stuff, and we talk about enhancers and silencers and those kinds of things, it's usually twisting the DNA around. So where your promoter was over here, you could have a silencer region hundreds or thousands of base pairs away, and it's blocking because it has something doing this and twisting the DNA around to physically attach to 2 points.
Okay.
Actually, what you see, the trip operon has some instances where it does that, and it's just and how the repressor shows up here, where it's twisting the DNA, and if it's a twisted up helix, you're not going to be able to open it up for the polymerase to be able to read it. The same way methyl groups being attached to a promoter are going to help silence it, because if you had a bunch of little methyl groups on here, those are physically blocking the polymerase from being able to read these sequences. And that's going to make it to where you can't read this gene.
Okie dokie, thank you.
On the topic of promoters, this is something, operons. There are some eukaryotes that have operons, but for the most part, we think about our genes being separated. Whenever you have a protein that needs multiple subunits to be able to form a functional protein, you can find those usually grouped as an operon. So I think a lot of our ribosomal proteins will be transcribed as one mRNA that then gets cut up into, okay, here's your 5S, here's your 16S, like those kinds of breakdowns. I actually might have flip-flopped, those might be bacterial. numbers. But either way, something where you need the multiple subunits before the protein can become functional, it makes more sense to say, well, if I can't use the one without the other, I don't need 200 copies of a 5S and 3 copies of my 16S. I need 50 copies of both. So by attaching them together so that you know that you have the same amount of mRNA for them, it saves you some of the trouble. But bacteria like to use operons because they want to have as little genetic information as possible. So cramming more genes together makes sense. And eukaryotes do have operons, but less commonly than how bacteria do things. Bacteria use operons way more often. All right, let's see. Okay. Number 7 was Lac Operon, the effects of lactose, glucose, major players. Well, I covered that. So, number 8 is general steps and players in DNA replication, transcription, translation. Pay attention to your strand directions. This won't be as emphasized as it was on the first one because now we have so much more to test you over. You might have 1 or 2 questions that'll throw back to be like, what was it? Which one was transcription? Which one was translation? And what was a polymerase? What was a ribosome? Just kind of like be able to talk about it at the level that I just did for that. I'm not going to go back and be like, what was TF2D? Like, what? Number 9, be able to write out DNA sequences to mRNA to amino acids. This was just like the question that you had on the first test. I believe that will make a comeback. Clear this up. If you are given a strand of DNA, let's say 3 prime, and I'm just going to make up some letters here. Okay. And now I give you a strand of DNA and I ask you for, give me the matching strand. So the matching strand here, swap it over to blue. If we're going, all right, if this is a 3' end, this will now be the 5' end. Bring it over a little bit. Probably should have spaced them a little easier, but if we have a T, we know it's going to match with an A, G to C, G, T, C, G, C, G, A, G. Okay. All right. So this is our complementary DNA. Okay. Now I ask you for RNA. If we opened up this double helix here, I should have had them separated, but we're going to start with reading 1 strand at a time. If we read the top strand and turn it into mRNA, we would have our 5' because we're always laying down 5' with the 3' butt open. So we're attaching to that magnet. Turning the top strand into mRNA, we would get A, C, G. We now switch to U, uracil instead of thiamine. C, G, C, G, C, G, A, U, U, A, A, C, G, 3'. Okay, this is 1 piece of mRNA. our other strand, so you have the option of going right here beside it, and knowing that it would normally be written down 5' to 3', I don't care which way you write it on here, just as long as you mark your 5' and 3' ends. So if the bottom strand is going to be in U instead of thymine. A, G, C, U, A. So these are the 2 strands of mRNA that would come out of this sequence, because we don't know which strand is being read. This would be part of a much longer chromosome. You would have it extending in both directions around it. So remembering that here. Chromosome keeps going in both ways. We just randomly grabbed a piece from the middle. All right, so we now have our mRNA done. I just realized I didn't give the start sequence. But we're going to pretend that there was a mutation along the way and we got an AUG, because I realized I can't do the next part unless I have an AUG in the sequence. So now to turn this into an amino acid, we want to find AUG. That's where we start translation. AUG, and that is an M. CGA. I don't have a codon chart in front of me. UUA might actually be a stop codon. But either way, you would have the chart. So then you would just say whatever this was, blah, 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 and keep going like that. Now, on the other strand, again, having to say that we had some sort of mutation here, so we can pretend that's a G. We always have to read 5' to 3'. Now, because it's written 3 to 5, we have to start reading this direction. And we come along, and now we have an AUG going this way, so we can start there and keep going. Thinking back to what you had on the first test, your answers, the longest one was I am smart. If you, and then it was a stop codon there. We don't know that that first M would be the one that the ribosome would start reading at, because again, you have extra signal codes. It's not just the first AUG it runs into. So it could be possible that it would actually start at the second AUG and it would have M smart or M art. And then when you read the other strand, there were, I don't remember what all the letters are off the top of my head. It was gibberish going the other way. And it was like MDLA, something like that. And it kept going. Again, I just made up gibberish on the screen, so I have no idea what this code's for. But when I say what are all of the possible polypeptide products, you could have the longest one. You could have, if there's extra M's, those could be potential start points. If you hit a stop codon, you stop. You do not keep going. Don't go, oh, here's a stop codon here, and then tell me that the next letter is an H. It doesn't work that way. That when you hit a stop codon, the release factors would attach and the ribosome disconnects. And then your justification for which one would be read, there's many correct answers. You could say the longest open reading frame, which is the one that runs off the end, or you could say the longest one that made a sentence that I could read. So in this case, I am smart, made a direct statement. if the instructions tell you to write the 1-letter code, meaning like the methionine gets shortened down to M, do it. There's one, it makes it a lot easier for me to grade because I can compare it to my answer key. And 2, if I put cute little messages in it and you write out MET for methionine, you don't get to see my cute messages. So.
I have a question.
Uh-huh.
So, you know how it says I am smart, and then there's another example that says M smart. If it starts with a start codon and there's no stop codon, and then it has like another AUG, it just continues.
Yeah.
Okay.
If it runs into AUG again, it'll just put in methionine.
Okay, that's what I thought. But when you were like, which one's the correct answer? I was like, I thought it was the first AUG, and then it just keeps going until there's a stop. But okay, that makes sense. That's just to kind of trick you a little bit.
Yeah, it's that when we have an entire strand, that 5' untranslated region might be fairly long before you actually find the exact one that it's looking for. It's kind of like how it's not every Tata box gets turned on. You have Kozak sequences, and there's all these different rules that the sequences are going to use that we haven't even uncovered all of them. And I didn't ask you to memorize them. So when I just hand you a strand, think about what the flu codon. There was lots of AUGs in there. Which one's your start one? Oh, crap, I don't know. Okay, let's go back and look at the amino acid sequence and then go, well, if it has to match this, then it must be this AUG that would start that sequence that we know is in the protein. So it gets a little squirrely. It's just because in real life, we have 3 billion base pairs. How do you know where a gene starts? You can't just assume every AUG is going to be a start. We don't know which ones are. And so we go, well, if this were the start codon, here's what it would say. If this one were the right start codon, here's what it would say. We just don't know. So you start it as if every AUG was a start codon, then you can say, well, here are all the possible polypeptide sequences that could come out of this piece of DNA. And then they would do further testing and figure out which one is the correct one. And realistically, if you have a gene that is 2,000 amino acids long, or if you have a gene of polypeptide that's 2,000 amino acids long, and you go, I am smart, stop codon, well, you know that's not the right one. So you would keep looking for a better AUG. And then you go, well, this one runs off the sequence that I have, but we know this is just a very short piece of DNA that I'm giving you. So. Any other questions about this? Okay.
Nope.
All right. All right. Lab skills, number 10. So restriction enzyme usage, gel electrophoresis, PCR, microarrays, etc. So going back, we use restriction enzymes back when we were cutting apart DNA, cutting it and putting it back together. That's where you had your sticky ends and your blunt ends. So you should still remember how to do a DNA map, or if I gave you a plasmid map, that you can remember how the sequences work. Sorry, the cut sites work. Let's say here is our map. We're going to draw. This is 0 slash, I'm making up numbers here, 4,000. And let's see, we have cut site eco R1 4,000, and we'll say this is at 700. Let's see, you have another one here. MH1 at 1,000. All right. So let's say you have a plasma map that looks like this, and I ask you, what size strands would you get if you digested with Eco R1? Now this is, think about this as a rubber band. If we make a cut here at the red one, right there, and we make a cut here, there are now two pieces, 1, 2 of DNA. The difference between 2,000 and 700 means this side of the DNA would be 1,300, just straight subtraction there. And we know that our total size is 4,000. That means everything else that is left over here, and you can write it out longhand if you want. I could say it's 2,700, but 4,000, and you know 1,300 is already used by this piece. Subtract it, 0, 0, 7, 2. So you get 2,700 for this big piece of DNA on the backside. So if we cut it with Eco R1, you don't put any other cut sites except Eco R1. You don't put a cut site at 0. You don't put a cut site at BAMH1. You only want to look and say, well, this one's address is 2,000. This one's address is 700. How many houses are in between it on the street? And you can just subtract. And then once you know one piece, you can just subtract that from the total to get the size of the piece that was left over. If we said, all right, now you cut it also with BAMH1 and Eco R1, that 2,700 piece didn't change. So it's still 2,700. But now we have a piece that is between 1,000 and 700. That means this is 300. And this piece down here, 2,000 and 1,000, is now 1,000. Which, if you think about 1,000 plus 300 added together, gives us the 1,300 that we had the first time.
And that's when it asks the size strands for Eco RI1 and for the BAMHI.
Yeah, that's a, if it asks for both, you digested it with both pieces.
Is there a possibility that it will just ask the cutting of just BAMHI 1?
Yeah, so if it said you cut it with just BAMH1, what size band would we get then?
Would you just...
It's if you took a rubber band and you made one snip in it. Now you have a line. How long is the line?
Would be 1,000?
It'd be 4,000.
Oh, 4,000. Okay.
Total size up here is 4,000.
Okay.
So yeah, you took this rubber band and you made one snip at BAMH1. That leaves 4,000 base pairs that are out in the open now.
Okay.
Yeah, one cut leaves you one band. 2 cuts gives you 2 bands. 3 cuts gives you 3 bands. So always make sure there's not extra, like BAMH1 in this case only had 1 cutting site, but Eco R1 had 2. So make sure you find all of the cut sites, but if there's like Eco R5, that is a different sequence. It has to be exactly what matches.
Okay. Thank you.
Okay, and remember, if it says something is undigested, that means it's still in its circle shape. It would be, in this case, undigested is 4,000. That last time when we had like the picture of the gel, and you had, all right, so you cut it, show me on the gel where the bands are, and then one of the columns asked for the undigested backbone, and a lot of people gave me like all these different cut sites. Undigested means it has not been cut. You should just have your original piece of DNA. And speaking of gels, since that was something, if you have your gel, remember DNA is negatively charged, so it's going to run to the positive end, that run to red. So I guess I can do it that way. Draw it big. And DNA goes this way. Smallest go the fastest. Smallest strands, largest strand. You would usually have your ladder. So if you had, bands that were at, draw L for ladder. You had 5,000, 2,000, 1,000, whatever. Numbers in between. And I said, okay, tell me the size of here. And then you went, okay, let me find the band for 300 and put a mark there. And then a band for the 1,000 is here. And then the 2,700. I'm just making up numbers here. And that was lane 1. Whatever. If I ask you for undigested, then it's just the one band at 4,000. And that's all you would need. PCR, you just need to know the general understanding that we use PCR to make copies of DNA, and it makes copies in the exponential way. If this is your original DNA here, double-stranded, the first thing we need to do is then denature it. Denature at 95 degrees. Remember, it's really hot. (95°C). Then we anneal our primers. So denature was step 1. Step 2 is anneal primers. Remember, these are just the little pieces of DNA. They're like 20 to 30 nucleotides that will attach to our sequence to say, hey, start copying here, because the polymerase can't start copying from single-stranded DNA. It has to go from double-stranded. So this gives us a little bit of double-stranded DNA. And then number 3 is elongate. Elongation. I can spell. And elongation means we're going to fill this in. And now the whole cycle can restart again with both of these splitting apart, primers re-annealing, elongation happening. So on every cycle, we end up with a doubling of the number of strands. And then it doubles again, and it doubles again, and doubles again. So you end up with billions of copies within 30 to 40 cycles, depending how much starting material you had. I'm going to call attention that on your primers, you would want to make sure that like that they are sequence specific. If it turns out that it could bind to a bunch of different genes that aren't the ones you were wanting to look at, so like the group that was looking at, or who had originally wanted to do their project on lactose intolerance, well, if you picked primers that could bind to genes that weren't just about the lactase enzyme, you wouldn't get the ones you wanted, or like you get a bunch of products that weren't necessarily what you were looking for. You want to make sure they are sequence specific. They should be about 20 to 30 base pairs long. That helps to make sure that it's sequence specific. If something is 20 to 30 base pairs, chances are it's not going to be repeated that many times in the genome, unless it's just a very, very repetitive sequence, and you don't want that. You want to make sure that it can't stick to itself. If you had like a palindrome, so remember back when we were folding our pieces of RNA and that you could stick them together, and if the sequences matched, if that was happening, you would end up with just little fragments here of being like bleep, bleep, and you wouldn't get really any elongation. So we call those primer dimers. You don't want to have primer dimers. You also want to make sure there's no long repetitions. Repetitions. Like A. For example, these are just ways that it can slip off. Also, you would want to have a balance of the number of C's and G's versus AT. Because of that, the whole ATs having 2 hydrogen bonds, CGs having 3 hydrogen bonds, and you need to have them be able to stick and break apart at the right temperature. So there's certain percentages, which are, it's more detailed than you would ever need to worry about for this, but you don't want it to accidentally be too hard to melt back off, because your temperatures need to match within a certain number of degrees from each other. And CGs take more energy to break because there's 3 hydrogen bonds compared to A's and T's. All right, any questions on this?
Nope.
Nope, all righty. Give me just a sec. All right. All right, linkage mapping. So this is for combination frequencies and creating a gene map from them. If there's any questions about this on there, it's going to be way more simple than the one that I gave y'all. Since I have a video up for that already, I'm not going to go through all of that again. But just remember that you're always looking for the ones that are different from the parentals. The number that is the most commonly occurring one is usually going to be the parental version. So, or the question will tell you, here are the parents, and here's what their offspring. So you want to look for recombinants, which are ones that are not like the parental ones. Mutations, you just need to know about the different types of mutations and what kind of impact would they have. So like a point mutation that is like a missense mutation, it's only going to change 1 amino acid. That's probably, that could be devastating for a cell, but it could also have no impact at all. Sometimes it's slightly beneficial. By comparison, if you have a frameshift mutation, this is a deletion or an insertion of more of 1, 2, however many nucleotides, it's not 3. Because if you drop out 3 nucleotides, that's 1 amino acid, and that isn't going to throw off all of the others downstream. Losing 1 or 2 or adding 1 or 2 is way more devastating. And if you have a change that occurs, it causes a frameshift mutation early on in the sequence, that's going to be way more devastating than one that occurs at the very end of the sequence. Any questions about this? There's not going to be any questions about like the ones that are tautometric shifting and anything like that. Maybe kind of know the types of things that, like general things like UV radiation is a physical mutagen, but not. I'm not going to give you like any chemical names and be like, what kind of mutations does this cause? You don't need that.
So when we did the decoding the flu example, that was kind of what it had to do with mutations, correct?
Yeah.
From what I can remember. Okay.
Yep, just like that, saying, all right, here's your strands, and we added 1 letter here. What's going to happen? Versus, all right, we changed 1 letter. Okay, well, now it's a nonsense mutation because it made a stop codon.
Okay, sounds good. Thank you.
And last but not least is gene expression. So ways to increase, decrease, prokaryotic, eukaryotic, RNAi. So we did talk about this a bit with the cell fusion case study. So you can go back and look at that. So ones that prokaryotes use are going to be things that are only usually at like the very first layer of inhibition. So remember, there's a nucleus involved in eukaryotes, and so they have to completely process their mRNA before taking it out to go meet up with the ribosome. That gives way more levels that modifications can happen at, where bacteria can take their mRNA and have ribosomes attached to it before it's even done copying down the mRNA. There's no nucleus to block it off. So that means there's no RNA processing to occur. And there's very little post-translational modifications to occur either. There's a lot of things that we do to our proteins to fancy them up that bacteria can't do. That was one of the reasons back when they first started making insulin for diabetic injections. We had to actually take it from animals or humans sources because bacteria weren't complex enough. Even if we gave them the right DNA sequence, they couldn't process it into a usable form. They ended up having to make a lot of changes to the amino acid sequence before they could start making it in bacteria. So by comparison, both of them can use things like repressors and activators and stuff, but there's just way more extra steps that can occur for eukaryotes. And in terms of ways to increase or decrease, things like obvious ones, repressor is going to turn things off. Activators are going to turn things on. But there's also those like double layers of, well, if you repress the repressor using a co-repressor, or if you're repressing the repressor, that's called an inducer, because then it's going to take off the repressor. Now it has induced the gene to turn on, versus if the repressor needs something to help it bind, that's a co-repressor. A repressor plus a co-repressor is going to silence more. If you have an activator, it can be blocked so that it can no longer bind to activate things. You can also talk about like methylation, silencing things, acetylation, making things more likely to turn on. And then that gets into like histone modifications. So histone modifications are something that we generally see in eukaryotes. not in prokaryotes. Not as much, at least. I mean, I actually have to double check if they even really have histones. They have different scaffolding systems. We didn't go into details on that. So, and how RNAi works, remember that's where you had like an anti-sequence. And if that anti-sequence was able to bind to mRNA that was floating around, it would make it double-stranded. And the cell would go, wait a second, I'm not supposed to have double-stranded DNA outside my nucleus. You must be viral, attack. And then it goes around and attacks any of that and cuts it up. And that's a way to get rid of mRNA before it can be turned into a protein. Shortening a poly-A tail is also going to turn down gene expression because remember there's always enzymes going Pac-Man style from the end. If it has less of a protective layer of A's, it's going to get chewed up faster. The longer an mRNA can sit around, the more times it can be read and turned into a protein. Okay, anything else?
I have a question.
Uh-huh.
So I know we just did that microwave lab, or I think I said microwave. I don't know. Anyways,
Yeah,
but what exactly do you want us to know from that?
You just need to know, like, if it's just a way to think about gene expression, that if you see an increase, you compared to a baseline, because you can't say there was an increase unless you had a starting point, of just being able to know, hey, certain genes are getting turned on, certain genes are getting turned off, and kind of the comparison there, if you're on looking at cancer cells, well, turning off a suppressor gene ends up with more more self-proliferation, or you could turn on a oncogene that causes more cell division. So it was just a way to practice thinking about how to turn things on and off. You're not going to be asked like very specifics on reagents and stuff like that. It's just for practice. I am also going to be recording and posting a video walkthrough of that lab because some people weren't able to get it to play properly.
Yeah, it's like it wasn't working on Chrome, so you had to go to the basic internet, the like Microsoft Edge or something.
Yeah, I tried to use Mozilla and then something else, and it let me download it, but then it wouldn't let me go on Canvas at all. So I was either stuck with no Canvas or no lab, and I was like, okay, I have the PowerPoint. So I'm just gonna have to watch the video.
Yeah, just really, yeah, I'll have that video posted tonight too, for y'all so that you can look at it. And it's, again, it was more for practice. And I do believe I'm gonna have I'll start the exam on Wednesday and give y'all 2 days to do it, Wednesday, Thursday.
Wait, really?
Is that okay?
Yes, I thought it was only going to be like 1 day for like 3 hours open or something. Because that's.
Well, I mean, you'll have a limited amount of time once you start it. It'll be one of those. Once you start it, you have to do it all in one sitting.
Right. I meant like some teachers are like, okay, at this time you have to. Well, my teachers at least. They're like, okay, you only have 3 hours on Tuesday night in order to finish it, or like 4 hours.
I think it'll be, I think she had it set up, it's either going to be an hour and a half or 2 hours. And I think it will be on either HonorLock or Lockdown browser. I think it's supposed to be on HonorLock. So I think HonorLock is supposed to be something that you don't have to download, but I do believe it requires a webcam. So if somebody does not have a webcam, it's basically you're being recorded while you take the test, and it makes it so that you can't open any other browsers. I'm going to double check on that because I haven't used it and I want to make sure. Now, if somebody has an issue like technically, like I don't have a webcam, something like that, get with me and we'll figure out a way to do that. And I'm going to post that information up for people. I'm going to just double check because the other teacher gave hers last week. But since her students had been using Lockdown Browser on all of their quizzes, she's like, I'm just going to do that instead. But since my understanding was that you had to pay for Lockdown Browser, and I wanted to make it to where y'all didn't have to pay for anything. So I was told that we could use HonorLock for high exams. Like, we couldn't use it for regular quizzes, but we could do 2 exams over the course of the year. And I'm going to use it for this one. And then the exam for your final next week is going to be over the board game you created. So you better get an A on that without needing to cheat. So I was like, I'm not going to bother like using school money and making people worry about webcam and crap for something that they like, because my average is usually around an A on that. I'm not gonna worry about it.
Are you gonna be at the school? Cause we did our like a physical board game. Are you gonna be at the school this week? Cause it says that it's opened today, but
so it was originally like they told us that we were going to open today, and then we're like, oh, you don't have to come back until May 16th. I'm gonna find out if worst case scenario, if whoever has the physical board game, I can meet up with y'all somewhere.
Okay.
Whoever lives, I live near 75 and 635, so I can go pretty much anywhere and just do a no contact pickup of pick it up off whoever's porch or whatever needs to happen. That I can do that.
Okie dokie. I was planning on probably I have to still talk to my lab mates because we have some questions that we have to do for the board game, and then we need to finish the video and a little bit of the paper. But I plan to have it finished Thursday, hopefully, so then I can also focus on the journal club too.
Yeah.
I guess I'll just email you about that whenever the time comes. It also depends on how things work out in the next couple of days.
Yeah. So yeah, just stay in communication with me, and we'll schedule a time to be able to pick it up. And most of the paperwork stuff you can submit digitally. It's only the physical. So if you have okay, well, here's the board, but now we have to do whatever else, and I'll have that to you tomorrow or whatever, then I mean they're being pretty lax on us for trying to accommodate schedules and everything.
Yeah.
So
What do y'all think about Texas reopening?
I think it's I am hoping if people were using proper social distancing and proper PPE, it wouldn't be an issue, but people aren't. And it worries me that now we're seeing a greater uptick in the rural area. By Lonnie. I don't know. My sister lives down in Waco and works at a restaurant, and she was already complaining that, I asked, well, are y'all wearing masks? And she's like, well, no, I'm not going to be the only one wearing a mask. I'm like, well, y'all are still, I was like, you're working at the cash register and talking to people, and y'all have an older clientele. You should be wearing a mask in case you're trying to explain to her about a. He doesn't need to say that word. And it's just been, I pointed out that if you can't get them to wear PPE when they're on full lockdown, if you lighten the restrictions, they're not going to suddenly start using more protection.
Right. Yeah. I don't know. I'm 19, and I see a bunch of people that I went to high school with, and even people that I work with that are my age, and they're going out to party, and they're going to the beach, and they're, Cinco de Mayo is coming up, and they're going to grill outs, and I'm like, wow, I have such a stupid generation.
Yeah, I live in Dallas off Katy Trail, and it is packed, and nobody is wearing masks.
Yeah, it's ridiculous. I'm terrified to go to Walmart without a mask and gloves because I don't know who's been there. I don't know where they've been.
Yeah. Now the gloves is kind of is if you don't have open things on your hand, you should be able to wash your hands afterwards. But if you're reaching and touching stuff and then touching your face is the main thing. So if wearing gloves reminds you don't touch your face, then absolutely wear gloves. But otherwise, regular hand washing is more than enough to kill this virus. And alcoholic hand sanitizer, if regular soap and water isn't available, is a good idea. But it's more about the not touching your face. And I've found that the pee pants analogy works well to explain how to wear the mask to people.
Pee pants?
So the idea is if you had 2 naked men standing there and one of them pees, there's nothing to stop it from going on the other person. But if the second person is wearing pants, there you have a little protection. And that's what wearing the mask is. If somebody pees on you, you have a little protection. However, if that first person is wearing pants and they pee, it just goes all over them and not onto that second person at all, even if the second person isn't wearing pants. Now, if both people are wearing pants, then it's almost impossible to get pee from one person onto the other without, particularly if you're staying 6 feet apart. So,
And then there's those people that are like, oh, I'm going to roll up my pants because they're uncomfortable.
yeah.
They cut out holes in their masks or they don't cover their nose. And I think it's really funny.
God, the number of people I've seen either mask upside down or they walk up to you and pull the mask down to talk to you.
Yeah, I saw a video of a lady. She was like, oh, it was so hard to breathe in these masks. And she literally cut a hole in the middle of it. I was like, wow.
Oh, God, I've gone out to Breckenridge Park a couple times, and I'll do full mask. Like I went out, I have a plague doctor mask.
Mm-hmm.
And we went for a walk in plague doctor mask, and then it looks.
That's awesome.
And they will stay 6 feet away from you if you look like a freak. So there's that. Also, if anybody does need cloth mask, I've been sewing them, and I have a nice pattern that's comfortable. If people want me to send the pattern or if they need to get mask, I can help provide them. Of course, because I have to do everything over the top. I have over 100 different kinds of fabric and stuff. So instead of cosplay, here's what I get to do.
Mm-hmm. It's funny. Maybe I should ask my sister to make masks because she does like cosplay too, and she makes a bunch of suits and her own clothes and stuff.
Yeah,
I could.
I use the pattern from Cricut, the like cutting machines. They have the pattern available, so even if you don't have a Cricut, you can cut. And they're like the flat front, kind of like curved ones that aren't the pleated monstrous ones.
Mm-hmm.
And it's been it's the most comfortable one I've found. So I've started making them for people. I have to actually that's one of the reasons I haven't been posting as much as I've spent the past 2 weeks sewing masks and crazy numbers. So anything to help. But all right, so if there's no other questions, I'm gonna go ahead and stop the recording. And then if anybody else wants to stay on or something, I just don't want this to.
I have a question about the recording for our PowerPoint for the journal article.
Uh-huh.
How would we do that? Because you said we don't have to video ourselves.
It's up to you if you want to do it like on a Zoom like this, where if you do a Zoom session and you say record, and you can do it as just hearing my voice or your voice. So I'm saying you don't have to physically stand in front of the camera and have it like projected on a wall. You can do it as showing the screen and you talking through as if you were in front of people. Mm-hmm. Or you can do it on your cell phone if you want to have a recording of it there and you're standing beside your computer, or however you want to do it. I think Zoom has been very helpful for when you do the recording. Then when you end the meeting, it will batch it up into a MP4 file, I believe. It'll give you like 3 options of here's just your video, here's just your audio, or here's the Zoom playback, and that's like them together. And then I've dropped it onto YouTube if you want to make your channel there, or I think you can just upload those files directly onto Canvas. Hopefully it won't ping us for size. I know that trying to email it to me could run into size issues. So I think there should be ways to post it online without needing to email it over.
And for the cell phone audio, we would just do, save email, upload the audio on the cell phone and the PowerPoint at the same time.
Yeah, I mean, you can do it that way. There's also if you do it on Zoom and you start the PowerPoint presentation and then hit the button to record your Zoom meeting, you can talk just on your computer, or you can do it and it'll batch it into one video, so you don't have to upload the PowerPoint as a separate thing, because we can watch the video of you talking through the PowerPoint.
Oh, okay, that makes sense. Okay, thank you.
For the time requirement, it says 15 minutes. I don't remember if you said it in the video or not. 10 to 15?
Yes, 10 to 15.
Oh, okay, that's what I thought. I was like, it's not like a minute within 15 minutes, because then I was like, that's a little hard
Like
to
your
reach for that. But
No.
10 to 15, that makes sense. Okay, cool.
Yeah,
cool.
10 to 15, and I do believe that YouTube will let you upload up to 15-minute videos without any authentication stuff. Though even if it tells you you must authenticate, it just means you have to tell it your phone number, and it sends you a text message, and you just and you type in the code. That's all authentication is to YouTube.
Oh, so it's not just
Real person.
videos.
Yeah.
Oh.
So yeah, and I do believe that's what because I already did it. I didn't run into that issue, but some of the other professors were talking about it, and they said, oh yeah, you just have to authenticate your channel to show that you're a real person, and then it'll release the time limitation.
Okay. And then we're gonna have to do responses for everyone, correct? Is that so? Like, are the videos gonna be all uploaded in like one place, and we watch them and we can comment
I'm
it?
gonna try to do. I guess I'm gonna post a discussion board and then have a discussion topic for each person, and then if you give me a link or a video or whatever, I can post it on there, and then people can do their comments straight on that. So it'll have everybody's like, okay, you click on. Be like, all right, here's Carolina's, and then they can follow your link and then type directly onto that. Wherever the link is of the video, it'll give a way to upload it.
Okay, sounds good.
And for the individual final, you said you would use the questions for our games?
Yeah, it won't be exactly your 10 questions, but it'll be topics about it and about how y'all's process of, what would you do if you had more time and money? Or, it's kind of weird because usually I would write these about the experiment that you did. But since I can't really be like, what was your independent variable? Like, what was your hypothesis? Doesn't quite work for a board game. So depending on the type of games that you give me, then I will base the questions. So it'll be over your game creation process and the topic you covered.
Okay.
So would it be like asking questions maybe about our rules?
Yeah, I mean, it'll be things that it should be easy for you to answer if you participated in the making of it.
And then are we going to play each other's games?
We were supposed to, I wanted to, but since the campus is closed, I mean, if somebody has it in a digital form, that I can do it. Now, if it's on a physical form, I can't, pass around the board game.
In physical form, like, we can't really play it. I wanted to have, like, maybe, like, 2 of my friends that live by and, like, test it on them.
Uh-huh.
Could we do that actually for our video instead?
Yeah.
Like, just kind of like a
playthrough
practice run, just so it's like easier to understand like how it flows rather than just listing out rules.
I love those like how to play videos instead of like trying to go through all the rules on new board games, especially on complicated games. They have like, if you just YouTube like how to play blah blah blah, it'll be like a little like few minute long video that goes through the rules and shows you the pieces and gives a quick like play example. Makes it a lot easier.
And for the exam, you said it'll be, when will it open again?
My understanding is that there have been at least police people and they could like unlock the buildings to let us in this whole time, but I don't know. I think they're trying to do rollouts on like the 16th or 17th. Let's see the calendar.
They're still doing classes online though for summer 1. I know that.
Yeah, summer 1 and summer 3, which is the long semester during the summer, are going to be online. They haven't decided yet on summer 2, and they've told us, if you have the capacity to work from home, continue to work from home. So they are trying to keep as few people on campus as possible. But I do think it'll open up more for like you to go in person to do some of the things. And I know that there were some campuses that had like Wi-Fi spots that were still available for students who could sit in their car and use the Wi-Fi.
Oh, okay. Well, that's cool.
So, but I don't know at what point they're going to open like the computer labs and stuff like that. I think it kind of depends on how much of a demand there is and how safe they can keep the people.
And our exam, that's on Wednesday. When will you open like the?
I'll probably open it on Wednesday morning and have it open, all of Wednesday, all of Thursday, and then have it close on Friday. So then y'all still have time to get your other stuff done because I have to have everything graded. My hope is that I can have it graded before Monday. And then you have your last test on Monday, which I guess will probably be Monday, Tuesday.
Okay. And then for the video of us for the board game, do we each take a video of us doing it?
So you can just do one.
Just one. Okay.
Yeah, it's a team grade for what you're turning in for the board game, and then your test over the board game will be an individual score.
Okay.
So is there going to be a separate like grade in the grade book for just the board game? I don't know if I saw one. I
Yeah.
saw the questions for the pretest and whatnot, but I didn't see like an actual grade for the board game itself.
Yeah, let me pull up the syllabus. My percentages. Okay. Yeah, so team activities, readiness.
And then we're going to be also doing the surveys over our teammates, correct?
Yes.
Okay.
So final exam that you're going to take next week over the board game is 10% of your grade. And then your team project overall is 20% of the grade, but your grade that your team gets will be multiplied by your peer evaluation.
Oh, okay. So if you get better reviews, you'll have probably a higher grade. And if you don't
Yes.
have as good, it's not
Yeah.
as good of a grade. Makes sense.
Yeah. So somebody who didn't contribute at all, even if your team got an A, you might still end up with a 40, because
That's fair.
yeah. And then somebody who went above and beyond can then actually end up, if your team ended up with a 90, you might end up with a 95 because you ended up with a little bit of a bonus on there. But that's why it's important to grade people with thinking that average is a 3 so that there's room to improve, versus if you go in with everybody gets 5s, then there's nowhere to go but down.
Yeah.
So, yeah. But usually, and I'm hoping on this, I haven't gotten any messages recently about anybody being like, my partner's completely MIA. So hopefully everybody contributed and we won't have any problems. So yeah, in terms of a bit paying attention, exam 2 is at 12.5% of your grade. Journal club is 10% of your grade. Final exam is 10%. Research project, which is now the board game, is 20% of the grade. So 20, 30, 40, 50% of your grade is due within like the next week or two. So if you're going, my quizzes aren't as good, don't worry, those are going to be, all of your quizzes are 7.5%. All of your homeworks are only 10%.
So for exam 2, we're not going to do like corrections or anything like that because that wasn't planned with exam 1.
No, but I think it'll be, you're going to see that there was some question. Oh, that does remind me, I need to upload.
Oh yeah, for the video tonight, when are you going to upload that? Because
don't worry about the deadline for the microarray. Like,
oh, okay, cool.
None of those are pinging off late points or anything. That was just like hey, have it done. But since I've been so busy, I haven't gotten the video uploaded, then that's not y'all's fault.
Oh, no worries.
Yeah.
Also for my group, we got one of the questions, like we didn't finish doing the diagrams for the Lac Operon. Are we still able to turn that in if we were to redo it?
Yeah, any corrections that are made, anything like that, go for it.
Okay, cool.
So, yeah, again, all of the team activities together is only 10% of the grade. So if any aren't super great, like your one exam 2 counts more than all of your team activities put together. So if you have to choose between what to focus on. Now that being said, the team activities were supposed to help prepare you for the exams. So yeah, looking over those should help. Like that's why, like, all right, if we did this in class on the flu codon or whatever, then me asking about it on the test won't be like, oh, which strain was the one that was bad? Like it'll be the same ideas of, okay, find the ATG start site, that kind of thing.
Okie dokie.
All right.
Well, that's all of my questions and everything. I just basically.
Okay, and I think we did a pretty thorough walkthrough. So
Thank you so much for the review.
Yeah, this was really, really helpful. I was big stressing.
so and sometimes it's just like it's been so long since we've used
Yeah. But honestly, when you were explaining it, I was like, okay.
You're like, oh yeah.
I know this.
Yeah, it's just a reminder thing. I'm gonna go ahead and stop